In this video, we're going to compare the mechanism of action between lipid-soluble hormones and water-soluble hormones. What we mean by mechanism of action is how does that hormone function in order to get that target cell to change. You're going to hear that phrase, mechanism of action, quite a bit, not only in anatomy and physiology, but also in a pharmacology course where you learn about how drugs work. When you hear the phrase mechanism of action in pharmacology, you learn about how that molecule either interrupts a enzymatic pathway in the body or possibly mimics an enzymatic pathway in the body. But in this context, we're just looking at how do these hormones work based on the fact that some are lipid soluble while other hormones are water soluble. So what we have here is a drawing. This comes from your book. It walks us through the mechanism of lipid soluble hormones. So what you don't see here is the organ or cell that produces the hormone. They show the hormone here as being this green structure, but somewhere prior to this it was produced by an organ. Since we're talking here about lipid-soluble hormones, let's choose, for example, from our list previously, let's choose testosterone. We've heard of testosterone. Testosterone is produced by the testes in males. It is a lipid-soluble steroid hormone. We could have just as soon chose estrogen or aldosterone or cortisol, but let's just use testosterone as our example. So it would have been produced by the testes, placed into the bloodstream. Now, based on the fact that blood is largely water and testosterone is a lipid, they don't solubilize. The testosterone molecules cannot solubilize in blood. So, lipid-soluble hormones require a transport protein. This transport protein, shown here in blue, helps that molecule solubilize in blood. Therefore, it can circulate throughout the cardiovascular system. So that's number one in our different steps here. Number two has also to do with the chemistry of that molecule being lipid. It can freely cross the plasma membrane of the target cell, shown here. So this is the target cell. It has a nucleus containing its DNA. It has a plasma membrane. And since this molecule is a lipid and the plasma membrane is largely lipid, phospholipids, this hormone can freely diffuse through the plasma membrane. So it can enter the cell, the target cell, where it will then find its receptor. The receptor for this hormone is intracellular, so it's found inside of the cell. Sometimes it's actually found in the nucleus. Sometimes it's found in the cytosol. It looks like this one is shown as being a nuclear receptor. So the receptor is found inside of the nucleus. Once it finds that receptor, it binds to the receptor, so they attach to one another. Then, very importantly, it has to find particular genes inside the DNA. So inside this nucleus, there's going to be DNA found in chromosomes, and certain genes are going to have the ability to be altered by this hormone receptor complex. And when the hormone receptor complex binds to the particular genes, it brings about genetic expression. Genetic expression has to do with transcribing the gene, so taking the DNA and converting it into RNA. If you recall, that's called transcription. Then taking that mRNA and translating it. Translation uses the mRNA code to make a protein. So ultimately, this process results in synthesis of new cellular proteins. These proteins are often enzymes that are used to produce reactions in order to allow that target cell to change what it's doing. Okay? So these are the four primary steps in which all lipid-soluble hormones work. The difference is the target might be different, the genes might be different, and therefore the cellular activity might be different. So we want to keep in mind these steps and compare them to how water-soluble hormones work. Water-soluble hormones don't require a transport protein. So you'll see in this drawing that the water-soluble hormone does not need any kind of transporter because blood is water-based, and if you're water-soluble, you can solubilize in the blood. So no need for a transport protein. Number two is the site of the receptor. Since these hormones are water-soluble, they cannot pass through the plasma membrane, which is made up of phospholipids. So the receptor must be on the surface of the target cell. The receptor is typically a membrane-bound receptor where there's a binding site for the hormone on the outside. So now that hormone doesn't have to go inside. It binds to the receptor, and then it triggers events within the cell. So binding of the hormone, which often the hormone is known as the first messenger, to its receptor activates events that ultimately lead to a change in what the cell's doing. We're not going to get into the details of these events, but it includes a secondary messenger. So if the hormone is the first messenger, the secondary messenger is often a molecule that's produced inside of the cell that triggers events within the cell. A very common secondary messenger used by many cells is known as cyclic AMP or cAMP. And once that cAMP is produced, what often follows are phosphorylation events. Phosphorylation means adding phosphate to proteins. And adding a phosphate to a protein often turns that protein on. That protein often is an enzyme that might be sitting inside of the cell inactive. So here we see a protein, an unphosphorylated protein that was produced by the cell, but it's not functioning. It's not turned on, if you will. These events prior lead to adding a phosphate group to that protein, shown here as the letter P. And now that that protein is phosphorylated, that protein is turned on, and those proteins that are phosphorylated end up producing the response. Notice there's no DNA involved here, so it's not a genetic expression change. It's more of a protein on-off change. So what I would like you to do is focus on the differences between how a lipid-soluble hormone works and a water-soluble hormone. The next thing I'd like to do is categorize hormones based on where they go. This is a little easier. Typically, we can categorize hormones by chemistry, lipid versus water-soluble, but also where do they go? Do they go far? Do they act at a distant target cell? Or do they work very close to where the hormone is produced? Most hormones we're going to learn this semester are circulating hormones. What that means is the hormone is produced by a cell. Here we have an endocrine cell. The hormone goes into the bloodstream and it circulates throughout the cardiovascular system. And then whatever cells have the receptor for that hormone are called target cells. And those target cells will then change. And you'll see that this would indicate that the hormone is water-soluble because the receptor is found on the outside or the surface of the target cell. Most of our hormones are circulating hormones. There are some hormones that act locally. So local hormones act in the same tissue in which they're produced. And then local hormones can often be divided into either paracrine, where the hormone acts at a nearby target cell, or autocrine, where the hormone actually is produced but also can bind to a receptor on the same cell that produces the hormone. There's a lot of hormones in the immune system that are local. They'll activate immune cells in a certain tissue as opposed to activating immune cells at a distant tissue. So circulating versus local hormones. The next topic, it might be a little bit of a review. I think we've talked about negative and positive feedback in AMP1, but this is often how hormones are controlled. This shows a basic negative feedback mechanism related to thyroid hormones. Basically, if you recall, negative feedback means that the response is opposite to the stimulus. So for instance, if there's very low levels of thyroid hormones, or we need to boost metabolism, all right, and we'll get to that a little later, but thyroid hormones basically boost metabolism. But let's imagine that there's a low level of thyroid hormones. Well, that's the stimulus, low thyroid, low T3, low T4. What that does is trigger events that will ultimately produce more thyroid hormones. And that's called a negative feedback. What this involves is the hypothalamus and the anterior pituitary gland. We're going to revisit that a little later when we get to the thyroid. But notice if you walk through the steps, a low T3 and T4 will stimulate the hypothalamus and then the anterior pituitary to stimulate the thyroid gland to produce more T3 and T4. If T3 and T4 get too high, they will then turn down the hypothalamus and anterior pituitary gland. So this shows a negative feedback here, meaning the stimulus now was high T3 and T4. The result is lowering T3 and T4 production. So that's what negative feedback means when it pertains to hormones. If the hormone level gets too high, we shut it down. If the hormone levels are too low, we stimulate the production of said hormone. There's also control that is positive feedback. Positive feedback is where the stimulus needs to be reinforced, or we need that stimulus to occur more. A great example is normal childbirth, which is hormonal. The stimulus is the baby is being forced into the cervix. The cervix contains the opening to get out of the uterus. So that's the stimulus, okay? Stretching of that cervix because the baby is starting to be forced out of the uterus. So a positive feedback would mean we want to reinforce that event. We don't want to keep the baby in anymore. So once the baby starts to be forced out of the uterus, we want to do that more and more and more. So we positively reinforce that event. So what happens is the uterus has stretch sensors in the cervix. And once that baby starts to push down on that cervix, it stretches it. That sends nerve impulses to the brain that result in the production of a hormone called oxytocin. Oxytocin then stimulates uterine contractions. And those uterine contractions force that baby down into the cervix even more, which stretches the cervix more. More stretching of the cervix positively reinforces more production of oxytocin. So notice that the result is also stimulating more of that stimulus to occur. So that's called positive feedback. There aren't many hormones that operate this way. Oxytocin is always the classic hormone that operates via a positive feedback loop. But we'll talk more about oxytocin later on. So this video, we should understand the differences between the mechanism of lipid hormones and the mechanisms of water-soluble hormones. We should understand the difference between a circulating and a local hormone, and then know the difference between a paracrine and autocrine hormone. And then the third major concept was the difference between negative feedback and positive feedback as it relates to hormone secretion. The next video, we're actually going to get into endocrine organs and the hormones that they produce.